the autonomic nervous system. ANS mediates many of our classical homeostatic processes, such as temperature regulation, glucose, especially in the blood, as well as blood levels of lipids, the plasma levels of oxygen, carbon dioxide, and even pH. The heart and blood vessels are also innervated by the autonomic nervous system and will help us to control and regulate blood pressure. This does it not only at the level of the heart, where it can control heart rate and contractility, but also blood vessels, where it controls vascular resistance. The kidneys are also under ANS-mediated homeostatic control. Things like pH, sodium levels and other electrolytes, osmolality, even total body water. All of these items are being constantly regulated throughout the body, and minuscule and large changes are occurring because of the autonomic nervous system. So what are the structural divisions of the ANS? These are important, and also it's important to contrast these to the somatic motor system so that you have a good feel for what the differences are. In the somatic motor system, we go right from the spinal cord to the muscle of action. So this is one nerve that goes from the spinal cord to the muscle. In the ANS, works different. The sympathetic nervous system, synapses in sympathetic chain ganglia or autonomic ganglia, and that travels up and down the spinal cord, innervating a multitude of various organs and blood vessels all at the same time. So while the somatic motor system really only innervates a muscle, and more specifically a few fibers in that muscle, the ANS can innervate large swaths of organs and organ systems. The parasympathetic nervous system usually has a synapse both in the spinal cord as well as in an autonomic ganglia that's located in close proximity to the organ or other item that it's going to be innervating. So that's a big difference between the sympathetic and parasympathetic, and that is where the autonomic ganglia are located. Autonomic ganglia are located in a close proximity for the sympathetic nervous system, and then for the parasympathetic nervous system, they're located close to the organ of action. That big difference is important because it means that the presynaptic versus postsynaptic nerves are going to be different lengths for the different divisions of the autonomic nervous system. Sympathetic has a short pre, long post, while the parasympathetic has a long pre, short post. So once you have the nervous system output to the receptor, what different neurotransmitters are being released and what receptors are going to be engaged. If we go back to our example where we're going to compare things to the somatic motor system, let's look at that first. So normally here again we have the soma or cell body in the spinal cord. The nerve will go the length until it innervates a few muscle fibers. It releases acetylcholine. It is a cholinergic nerve. That acetylcholine leaves the axon terminal, goes across the synaptic cleft, in this case the neuromuscular junction, and binds to a nicotinic type 1 acetylcholine receptor that is ionotropic, meaning that it allows ions to travel once the acetylcholine binds to it. This is a very classic way of signaling. Now, the autonomic nervous system utilizes a little bit more complex of a process, so let's compare and contrast. If we take the sympathetic nervous system first, you have your first spinal nerve, soma, and then it will synapse in an autonomic ganglia. This autonomic synapse also uses acetylcholine, so this is the presynaptic nerve. It releases that acetylcholine, goes across the synaptic cleft, and binds to a nicotinic type 2 receptor. These particular receptors also allow for ions to travel through and will stimulate another nerve to be released. That other nerve to be released can be one of two types, either another cholinergic nerve or a noradrenergic nerve. So let's go through the cholinergic nerves first. Usually in this case, that cholinergic nerve will synapse and release its acetylcholine, go across the synaptic cleft and bind to muscarinic type 3 receptors. Now these muscarinic receptors are G protein coupled receptors. Rather than letting ions through, they're going to stimulate or cause a transduction of their signal via G protein and then stimulate something else, such as allowing for calcium influx, changing the amount of phospholipase C, so forth. Norepinephrine, or a noradrenergic nerve, can also be part of the sympathetic nervous system response. These are again postganglionic nerves that release norepinephrine into the synaptic cleft. They have a plethora of receptors to bind to. They can bind to alphas or betas, and there's a number of different types of each of these receptors. There's alpha 1s, there's alpha 2s, there's alpha 2Cs, there's beta 1s, beta 2s, beta 3s, and they will be at different locales in the body depending upon the density of which they are. Then they will get a metabolic response, vascular response, or whatever change that particular organ will undertake. So that's the noradrenergic pathway through adrenergic receptors, whether they're alpha or beta. But remember, there are a few examples of cholinergic sympathetic nerves. Finally, we use our last compare and contrast by looking at the parasympathetic nervous system. Here, the preganglionic nerve, remember, is longer in nature, but it still uses acetylcholine. It releases acetylcholine, and that goes across the synaptic cleft and binds to a nicotinic type 2 receptor. From here, that stimulates another nerve, and that second nerve is also cholinergic. So then this postganglionic nerve is cholinergic, releasing acetylcholine into the synaptic cleft. This can bind to a number of different types of muscarinic receptors. M1s, M2s, M3s, and there seems to keep increasing the amount of M's they want to denote. But this will engage things like smooth muscle, glands, various types of processes in which you would want to change from the parasympathetic nervous system. These are G protein coupled receptors as well. Therefore, when they get stimulated, they will stimulate a G protein that will then do a particular action, like activate an enzyme or open a channel. If we compare and contrast all of these, we'll notice some similarities. And the first similarity, acetylcholine is also always the first nerve in this system. The first synapse is always nicotinic. In the somatic, it's nicotinic type 1. In the sympathetic and parasympathetic, it's nicotinic type 2. This is then where things start to change after the first synapse, because now either you could have a cholinergic or adrenergic nerve, and then of course the downstream receptors, there are quite a few of those. Another difference with nerves are in the ANS, axon terminals are a little bit different. Instead of being a single axon terminal, where it kind of buds out and has a nice synapse with one particular locale, axon terminals in the ANS are oftentimes in bead-like strings. This allows for more widespread engagement of the peripheral tissue rather than just a single response. These particular axon terminals, also termed varicosities, will allow for more neurotransmitter to be released across that range of tissue.